Good afternoon, grade 12s. I'll get you to copy down your learning objectives and your success criteria while I mark the roll. Okay, so before we start our lesson today, we're going to go back to what we did in yesterday's lesson, where we started our new unit, Hereditary and Continuity of Life, which is the start of your final unit 4 before you're finish up for the year. Let's just go to our review. So yesterday we started looking at what DNA is and what its function is, okay? Today we're going to go into a little bit more of its structure. But just going back to last lesson, we particularly looked at this diagram here, where we looked at how the DNA is bound up to these proteins called histones into nucleosomes, okay? And then coil and then supercoil, condensing into those chromosomes which we find in our cell. So remembering that the DNA is holding our genetic information, okay? And as the weeks progress, we're going to look at what happens with that genetic information and how it transforms into proteins. We also looked yesterday about the differences between our prokaryotic and our eukaryotic cells. Remember, the main takeaway there is, and we drew this diagram into our books, is that the prokaryotic DNA is circular, and because there's no nucleus in those cells, it's just found in that cytoplasm. And sometimes we can be quite specific about that and say it's in the nucleoid region. All right, whereas in those eukaryotic cells, we have got that nucleus and it's found in there, and we can also find it in chloroplasts, in our plant cells, and also the mitochondria as well. And remembering it was linear in those eukaryotic cells. Okay, so that's just a bit of a sum up of what we did yesterday. We're going to do our quick 3-question review on that. So if you just grab out your pens, I've got 3 quick questions there for you. 1, what does DNA stand for? 2, name those 3 places where DNA is found in the eukaryotic cells. And then that final one is, what is the protein that packages and orders our DNA into those structural units that we call the nucleosomes? So I'll just give you another 30 seconds just to write down those couple of words, and we'll see how you go. All right, Hannah, what did you have for number 1? Yep, that's right, deoxyribonucleic acid. And so today we're going to start to break down what that word is and how it's showing us the components of what DNA is made up of. Henry, what did you have for number 2? Yep, so we've got the DNA in eukaryotic cells in our nucleus, mitochondria, and the chloroplasts for those plant cells. And Max, what about number 3? Yep, that's right, we've got our histones. So that's just giving you some basic content from yesterday, just a quick revision. Remembering I had your study resources up from what you should have gone through last night. And if you're still struggling with some of those concepts, we can look at it in this week's tutorial. So going on to today's lesson now, we'll just go back to our learning objective and success criteria that's written in your book. So today we're going to look at recalling the structure of DNA, including nucleotide composition, our complementary base pairing, and hydrogen bonding. So we're looking at the structure. So what does DNA look like? What is it made up of? And here are a couple of the things that we're specifically going to look at. Success criteria for today. First of all, I can label a diagram of DNA and identify its main components. Our first activity today is working through our worksheet, where we're actually going to look at what DNA looks like and what is making up that structure. Second success criteria is I can describe the structure of DNA using correct terminology. So it's all well and good that you can label a diagram, but you also need to be able to use words and write a paragraph to describe that structure and then what DNA is also made up of, using our correct terminology. And this is a big thing for today's lesson. We're looking at that scientific literature that we've talked a lot about, okay, where we can use scientific terms to describe what we're talking about. Okay, and then that leads us into our third success criteria. I can pronounce and spell key terminology from this lesson. So today we're going to introduce a lot more new words, just like we did yesterday, And we're going to make sure we can spell these terms correctly, and we can also pronounce them correctly and use them in the right context and be able to do that with some confidence. Just before we go into our lesson content, I just want to touch on those key terminology that we've got here, and I've made a separate tab on your OneNote so you can see the list of words we're working with today. As our success criteria stated, you need to be able to pronounce, spell and define these terms. Okay, I'm not going to go through all these terms now. We'll revisit them when we look at our worksheet. So just starting, I'm just going to do a quick brainstorm to start looking at this structure now. I want to find out as a class, what do we already know about the structure of DNA? We spoke a little bit about it yesterday. You guys did back in year 10, which I know is a couple of years ago now. You did your genetics unit, which touched on a couple of these things. So I just want, as a class, if we can just start imagining what a DNA molecule looks like, okay, what is it made up of? So let's start getting some ideas. Yep, Logan? Yep, you're right. So a molecule is quite small. We know they're fitting within a cell, and we know cells are quite small as well. Yep, Ty? Okay, yep, so you're saying it's got a spiral shape. Right, can anyone expand on what Ty's saying there and try and use some more correct terminology instead of spiral shape? Yep, Justin? Yes, we've got that word, we touched on it yesterday, double helix. And I really want you to be able to describe this molecule as a double helix rather than saying a spiral shape, okay? Who else have we got? Sophie? Yep, okay, so DNA is made up of genes. Yep, and you might be familiar with some letters that look like this, A, T, T, C, G, G, C. You would have touched on that in year 10. Today we're going to talk about what those letters stand for and then what their role is as well. All right, so now start to think, what if you were to describe that molecule, what does it look like? Yep, Hannah? Yeah, okay, that's thinking about it at a really basic level, but you could say you've got two strips down the side and stuff in the middle. Okay, so you're thinking you've got those two things down the side, and then that's what gets twisted around into that double helix shape, plus your stuff in the middle. And that's right, Ty, that's a really good suggestion too. It looks like a ladder. And sometimes you'll find in those junior science textbooks that you've looked at so far, that that's how it sort of describes that shape. Similar to what Hannah said before, you've got your two strips down the side, and then your rungs of your ladder are that stuff in between, in the middle, okay? And then that's the flat version of it, and then it moves into that double helix once you start twisting it up. All right, so I think we're starting to get a bit of an idea where we are at as a class in terms of what we already know. I just added some images down here just to start to get some images in our mind. All right, here we've got an artist's impression, and you can start to see the molecules that are running down those strips along the side, plus those rungs that are really clear. Unfortunately, at our school, we don't have a DNA model for you, but you might have seen them on YouTube videos or on TV shows, and you can start to see the complexity, and that actually looks quite messy there, but there is a definite structure to it, okay? And then over here, we can start to see what these rungs are looking like in between, and we've got some different colors representing some different things. Okay, I'll get these worksheets handed out now, and we're going to start to go through the different components and looking at this key terminology.